Hello, all of you summer students, and welcome to this lecture on cytology. So we're going to look at cells. That's how we always start with these classes, right? Looking at cells. I wanted to start with cancers, actually, because that's a really good context for us looking at why it's so important to look at cells and how it is that we think we know so much, but actually there's some dilemmas that we haven't solved in this world of science, right? So let's think of cancer. What is it? It's a tumor. It's a growth. It's a neoplasm. It's something that has grown out of control. It is mitosis gone wrong. It is cell division that happens over and over and over again when it shouldn't be. And this picture here is a liver that has all these growths on it. So cells truly are the basis of it all. So when this rate of cell division exceeds cell death, this is what we have. We have a growth. We have some sort of tumor. And it could be malignant or not, right? But that's just what it is. Mitosis gone wrong. Cell division gone wrong. So we know that it's a mutation. It's caused by a mutation in the DNA. And somehow that cell signaling of, all right, you have enough new cells, let's stop, continues on. And it continues on and starts to wreak havoc on our bodies. So some terms that that come up when we study all this are, an agent that causes a mutation in that DNA is a mutagen. So what are the mutagens? I mean, we don't know. If we knew them all, that we'd have solutions to this too. A particular agent that induces a cancer, right? Not just some little growth, but a cancer is a carcinogen. And another term, oncogenes. These are mutated genes that promote cell growth. So this is referring to the gene itself, an oncogene. And then we have, very importantly, tumor suppressor genes. Isn't that amazing? In your body that inhibit cell growth. So that you do have a gene that says, oh, wait, too many, let's fall back. And those are going to promote a DNA repair also. And so something gone wrong with that system can cause some of these growths. And some of these tumors can kill. A very interesting story is that of Henrietta Lacks. She died of cancer, and she had these cells that were overproducing mitosis. They were creating growths. And some of these cells were taken from Henrietta into these cancer study labs, research labs, and they looked at these cells and they found that they had this inherent ability to duplicate and duplicate and duplicate again. So they've been a wonderful source of study for research. And then that one lab sent it to another lab that sent it to another lab, said, look, these cells, these are great. We're doing cancer research. Look how great. And they were sent all over the world, have Henrietta Lacks's cells, and she's no longer living. And there's a very interesting YouTube here that I suggest you look at that talks about the problems that came with that. Did they ask her for permission? Did they not? Was her family remunerated for that? And so very, very interesting story about how some cells can live on and on. Amazing. So to look at cells and shapes, we do this in the beginning of our anatomy course where cells can be flat. They're squamous. They can be cuboidal. They can be columnar where they're long. They can be polygonal. They can be stellate. So, when you start looking at this, you realize, cell, like we look at the round cell with the nucleus in the middle and the mitochondria, isn't it really not all like that? There's an incredible variability in cells, and they make up so many different tissues. So this one, this fusiform, for example, is making up muscle tissue. This discoid is your blood, and we'll be able to encounter those as we move along in this class. Size. What about size? Well, we know that the cell is a structural and functional unit of an organism, right? We talked about that before. If you go smaller, then it's no longer living. They vary a lot. There's about 200 different kinds of cells. Most are teeny, 15 Microns, thousands, right, of a 1/1000th of a millimeter. And this is up here to show you what a millimeter is. So in centimeters, these are 10 millimeters. So most are very, very tiny. And the longest nerve cells that have been discovered are about 1.37 meters. So that's really when you're looking like a nerve cell and the axon goes on and on. So the long axon is what's making it very long, but the cell body itself is quite tiny. Lots of research has been done on the giant squid, and that's because it's an easy neuron to remove. They're not that huge. They're about 0.6 millimeters, but still much easier than other tiny, tiny neurons. Because to study these, just imagine how difficult it is to study. And then I always like the ostrich egg because it has the greatest volume. It's so big. I had to buy one once. I had an ostrich egg. They're just impressive to think that it's just one cell. So a cell has two major subdivisions, and that is the membrane itself, right? And what's inside, and the cytoplasm. So we can call it plasma membrane when we're talking about the cell membrane, and then everything inside. Okay, so first let's look at these terminology that can be so confusing. ICF, intracellular fluid. ECF, extracellular fluid. Everything that's outside of the cell. But this is a problem. We use more than one word for that. What are the other words? Tissue fluid. Tissue fluid is the same as ECF. Extracellular. It's outside. It's in the tissue. What else? Oh, interstitial. We use that a lot. Interstitial fluid. It's the same as ECF. It's outside of the cell. It's not within. Okay, all three of those terms are used always. So commonly. So it's something you do have to know. So I am introducing to you Joe Cell. Joe Cell looks like that, and he's rounded, or she, and has all these structures in there, and sometimes they have cilia, and sometimes they have microvilli, and this is kind of what they look like. But I'd like to stress that that's not what a lot of them look like. So first I want to start with the membrane and how it is. The membrane is crucially important. That's what's going to let things in that should come in, let things out that should come out, and not let things in that shouldn't and out that shouldn't, because if they do, you could pop that cell. You could kill that cell. So this fence is very important. So how do you build a good fence? Ingredients. 2 layers of lipid. So it's like a recipe. 2 layers of lipid. You're going to leave it. It's called a bilayer, a lipid bilayer. A handful of proteins. Where are the proteins on my picture here? So lipid bilayer, protein in purple, protein in red, protein. All these are proteins embedded in that layer. Handful of proteins, pinch of cholesterol. Cholesterol tends to be yellow substance here in this region, just around the center here. Pinch of cholesterol, a dash of sugar. This poking out in purple here, this is carbohydrates. And they assemble in a double layer. This is really what it looks like. So you have the little head and tail, head and tail, head and tail, all in this very long, long chain. I'm going to try and play this right now. Yeah, that won't work. I'm going to leave this for you to see, and you can look at a very short video on how that membrane behaves and why we call it the fluid mosaic membrane model, because it just sort of floats around. But because it's assembled the way it is, that floating sticks together. We'll get to that in a little bit. So this lipid, this is the structure of that phospholipid. And it looks like, this part here, we're going to just summarize all this complicated atoms that are in here and molecules into a head. And we're going to summarize this fatty tail here into just two tails. And that's why you see it as a ball and legs so often, because it'd be hard for us to go through every one of these little atoms every time to try and explain what's happening. But really, what is happening is that it has this phosphate here, this phosphate head. Okay, this is the phosphate part here. And I'm going to skip this here. And then it has glycerol, which is a 3-carbon chain. And then it has the fatty acid tails. Okay, and so heads here and the tails. These are all in a row and pointing both out of the cell and pointing both into the cell. Let's do a clicker here. I like clickers. The fluid found in the region outside of a skin cell, I just threw skin in there, could have been anything, is called ECF, ICF, interstitial, or A and C are correct. And in this case, D, A, and C are correct. There's more fat in the membrane. There's cholesterol. And we always think of cholesterol as such a problem, but we need it in the membranes. It's going to give it flexibility of that membrane. So 20% of the fat, of the lipid in the membrane is going to be cholesterol. Is that bad? And the answer to that question is no, it's not bad. You need it in there. We also have sugars, and those are glycolipids. So sugars attached onto a lipid. About 5% of them will do that. Sugar attached onto lipid in the membrane. They occur only in the extracellular fluid. So in my diagram here, you see them pointing out in green here. And they don't point into the cell, only to the extracellular fluid. And they're very important in how cells interact with each other. So there's the membrane with proteins, lipid bilayer, cholesterol, another protein. This is a glycoprotein, so you have sugar on a protein, and then there's sugars on fats, right? Lipid proteins. We're going to stop on this one. The head of this molecule is hydrophobic, hydrophilic, both A and B, or water repelling. So actually, see if you remember all this. Let's go back. If the heads are pointing to the outside, and the outside is full of water, and the heads are pointing to the inside also, and the inside is full of water, then those sections have to be water-loving or water-hating. They have to be hydrophilic because they're immersed in water in and they're immersed in water out. Okay, so before we look inside the cell, we're going to stop this and I'll record another video for you. Thanks for listening. Bye, guys.